Hi, my name is Alex Marshall. I am an assistant professor at North Carolina Central University, and today I'm going to talk a little bit about the reciprocal relationship between cytokine dysregulation and binge alcohol consumption. And so just as a brief overview of what I'm going to go over, there'll be a little bit of background about alcohol abuse and dependence, and then I'll talk a little bit about neuroinflammation versus neuroimmune responses. So you'll see a lot of times where I'll compare two different aspects of either alcohol abuse versus dependence or neuroinflammation versus neuroimmune responses, and then just go through a few results where I looked at a couple of cytokines and glial cells, and then end with some conclusions. I like to call them the take-home message. So alcohol abuse or alcoholism, as most people typically call it, is a huge problem in the United States. Approximately 10% of the population suffers from alcohol abuse or would be classified as having an alcohol use disorder. But over 60% of individuals actually consume alcohol in some form or fashion. More than 50% of Americans, because of this, because of the epidemiology around it, have a close relative who has or has had a problem with alcohol abuse. Estimates suggest that it costs the United States approximately $224 billion to correct issues associated with alcohol abuse. And that includes things like missing work, property damage due to car accidents, as well as healthcare from hospital visits. The vast majority of these expenses are associated with binge drinking, and I'll define what binge drinking is a little bit later in this presentation. You should know that 1 in 6 U.S. adults in the United States binge drink more than 4 times a month. And again, I'll define what binge drinking is a little bit later. But what my lab is interested in, or what I'm interested in, is what happens as someone goes from experimenting with alcohol. So maybe someone's a social drinker and occasionally they might have a glass or two, to when they start to abuse alcohol. And so when we talk about abuse, we're talking about alcohol under problematic situations. So whether it is binge drinking or drinking during periods that are inappropriate. So if you're underage drinking, that would be considered abuse. Individuals who are pregnant and drinking, that would also be classified as alcohol abuse. But binge drinking itself is characterized as drinking 4 to 5 drinks within a 2-hour period, and it results in a BEC above, or blood alcohol concentration above 80 milligrams per deciliter. And so we'll focus in on binge drinking as a particular part of abuse, but my lab is actually interested in what occurs during binge drinking compared to what happens when someone becomes addicted. So there is a progression that happens. There's changes in the brain, and there are changes in the peripheral system that promote alcohol consumption over time and lead to dependence or addiction. So while most people may not think about alcohol abuse as being a neurodegenerative disorder, people who suffer from alcoholism and are chronic consumers of high levels of alcohol do have damage to the CNS that's just as debilitating or damaging as individuals with Alzheimer's disease. And so you can see on the red line a group of people who their MRI was checked, or their brain was checked with an MRI, and you can see that it is just as damaging, alcohol is just as damaging as Alzheimer's disease in certain regions. On the right, there is a social drinker versus an alcoholic. And you can see that there are changes in the ventricle sizes. So there's an increase in the ventricles as well as a general loss of brain matter. And so my interest in the neuroimmune system originally started because of changes associated with damage. So when you think about the neuroimmune system, people often consider how neuroinflammatory responses lead to neurodegeneration. And so this is true in multiple sclerosis, it's true in Alzheimer's disease. And so what people have found, or what clinical studies have shown, is that alcoholics actually do have symptoms or signs of having some kind of neuroimmune response. And so on the right, it says IBA1, and IBA1 is a marker for microglia. And so at the top, there is a control a more social drinker versus at the bottom, someone post-mortem who was an alcoholic. And you can see that there are drastic changes in the microglial size. To the right, it's showing both MCP1, a chemokine associated with a neuroimmune response, as well as changes in receptors, so toll-like receptors. And you can see that for comparing the controls versus an alcoholic, there's a drastic increase in the concentration of either chemokines associated with a neuroimmune response or the receptors associated with a neuroimmune response. And so this all came out of the Fulton Cruz's lab down at UNC. And so this is kind of how I originally got into studying the neuroimmune system in terms of what happens with alcohol abuse. And so the reason for that is because when people, again, think about the neuroimmune response, they think about it in terms of being either a protective mechanism, kind of fighting against pathogens or damage to the system, or a housekeeper, so kind of getting rid of cellular debris from the damage. And so the neuroimmune response consists of glial activation, so microglia and astrocytes. The neuroimmune response includes the neuroimmune system includes the blood-brain barrier. So a neuroinflammatory response would include disruption of the blood-brain barrier, and a neuroinflammatory response would include infiltration of peripheral lymphocytes and neutrophils. And so again, at the top on the right, I'm showing some of the damage associated with chronic alcohol abuse. And so one of the theories behind that is that you have damage to both the CNS and the peripheral system that causes glial activation, disruption of the blood-brain barrier, and then infiltration of peripheral immune signaling. And so this particular graph at the bottom is from an Alzheimer's paper, but it's showing how peripheral immune responses can influence the neuroimmune system and cause degradation. And so that's one of the theories behind alcohol use disorders and neurodegeneration. But as I got into the neuroimmune system and really got interested in what was going on, I started learning more and more about this field called psychoneuroimmunology. And so in psychoneuroimmunology, the interest is more about how the immune system affects behavior or how it affects neurotransmission. And so this particular diagram on the right is from a paper on pain. But what's important about it is that you can see that there are cytokines that are secreted by activated glial cells. So again, we have microglia and astrocytes that are secreting these cytokines. But when they secrete these cytokines, not only can they cause an immune response that would be associated with neurodegeneration, but importantly, you can see that the neurons themselves have receptors that can respond to these cytokine signaling. Particularly what interested me is some of the pro-inflammatory cytokines actually map onto the pharmacology of alcoholism or alcohol abuse. So where you can see IL-1 beta and IL-6 actually inhibit GABA and can increase the glutamatergic tone. That's that actually maps onto what alcohol does. So alcohol is a GABA agonist that can also be an antagonist at the NMDA receptors. And so what really interested me is if we are seeing changes in the neuroimmune response and alcohol dependence, when does that happen? Does it happen before someone becomes dependent? And if it happens, does it affect behavior long before it affects degeneration? And so again, one of the first questions that I wanted to look at was what happens to the neuroimmune system in the transitionary period in an alcohol use development. So what happens when someone is binge drinking? So I don't know this guy, I'm going to call him Sean, but Sean is the epitome of binge drinking. Remember, I said that it is drinking 4 to 5 drinks in a 2-hour period, and he is going well above that and having what looks like 5 beers, and I'm assuming under 2 minutes. And so the way that I study that is by using a paradigm known as the drinking in the dark model. And in this model, we give mice access to alcohol for 4 days straight. 3 hours into the dark cycle, they are giving access for 2 to 4 hours, depending on the paradigm. But importantly, when they have access to this alcohol, they consume it themselves. They achieve blood ethanol concentrations that map onto binge drinking. So they achieve blood ethanol concentrations around 80 to 120 milligrams per deciliter. And they start to actually exhibit behavioral intoxication similar to that which you would see in a human. So they have motor deficits, and they actually consume this alcohol for the pleasure of consuming alcohol, as opposed for the caloric need associated with alcohol itself. The other important part about this model is that these mice that drink in this period are not dependent, and they don't show evidence of neurodegeneration. So we can look to see what's happening to the neuroimmune system way before someone, anthropomorphizing, way before someone is actually dependent on alcoholism or on alcohol. And so the first thing that we do is characterize what's going on to the neuroimmune response. And so we do that by using PCR, immunohistochemistry, and ELISAs. And so you'll see in the experiments that it's set up so that they either got alcohol once or 3 times, depending on the paradigm, or they got alcohol and then we waited to see what occurred in abstinence. So in the PCR experiments, they got alcohol over the 4 days, and we gave them 0 to 7 days of abstinence to understand what was occurring in the system, or they got alcohol 1 or 3 times, and again, depending on the experiment, were euthanized at different periods to understand the persisting nature of those neuroimmune changes. And so one of the first cytokines that we looked at was interleukin-1 beta. IL-1 beta is a pro-inflammatory cytokine. And so one of the first things that we saw using immunohistochemistry was that there's an increase in IL-1 beta in the basolateral amygdala. And so this slide shows on the left animals that got water or ethanol, and on the right, the animals either got water or sucrose. The top half is looking at the basolateral amygdala, and the bottom half is looking at the central amygdala. And so what we found was that specifically in the BLA, that there was an increase in IL-1 beta after 3 cycles of abuse. This did not occur when the animals only had access to sucrose. And again, it did not occur in the central amygdala. When we look to see how long it persists, because of the methodology, this is the entire amygdala as opposed to basolateral versus central. But what we found is that using PCR, ethanol exposure actually causes increases in IL-1 beta mRNA expression that persists even 7 days after abstinence. So where it says 0, that is immediately following ethanol exposure, as opposed to 7 days out, we still see that increase in IL-1 beta. Looking at an anti-inflammatory cytokine, IL-10, or interleukin-10 in the amygdala, we actually found the opposite reaction. So as the animals had more access to alcohol, they had a greater depression in IL-10. And so again, this particular graph is using ELISAs. And so what's important to point out here is that it only occurs in the CNS and not in the peripheral system. So at the top, we are looking at graphs of IL-10 specifically in the amygdala. Again, it's the entire amygdala, not BLA versus CEA. But you can see that after 3 weeks, you see a decrease in IL-10 following ethanol exposure. This did not occur with sucrose. But importantly, when we looked in the peripheral system and used serum, there were no differences in IL-10 between the animals that had ethanol or water. And so that kind of characterizes what happens to the neuroimmune system from alcohol abuse. So we have this pro-inflammatory milieu that occurs following ethanol exposure. But what I really want to understand is what's the relationship in the opposite direction? So how does this neuroimmune response affect alcohol abuse and some of the decision-making of the mice in order to consume alcohol? And so previously, there's been some studies from the Harris lab out in Texas showing that peripheral immune modulators actually affect ethanol consumption. And so they used PDE4 inhibitors and saw that it reduced ethanol consumption But what I wanted to do was to manipulate specifically the neuroimmune response. So without changing peripheral immune responses, if we can narrow in on the neuroimmune response and see if it actually affects consumption. In order to do that, we use stereotactic surgery and use guide cannulas directly into specific brain regions. And so this diagram is showing the cannula going into the central amygdala, but if you move it more laterally, then you would be in the basolateral amygdala. So we gave the animals access to alcohol to establish a baseline consumption. They underwent surgery, and then in a Latin square design, they either got the cytokine of interest or placebo or vehicle, rather, directly into these brain regions to see how it affected ethanol consumption. And so because we first saw the change in IL-1 beta, we originally started with IL-1 receptor antagonists. And so the IL-1 receptor antagonist directly into the basolateral amygdala actually reduced ethanol consumption. And so you can see in the bar on the left, that's our saline control. And on the right, those animals received IL-1 receptor antagonist directly into the basolateral amygdala. Importantly, this actually reduced ethanol consumption. I'm sorry, not ethanol consumption. It reduced blood ethanol concentrations. And so the animals fall well below what is defined as a binge level BEC. So remember, the binge level BEC is anything above 80 milligrams per deciliter. That's where you start to have a lot of motor deficits and the more classical intoxication parameters that are problematic in terms of driving and or interacting with individuals. And so we're getting BECs that fall more into the social drinking level. It did not affect sucrose consumption. And so when you give, when you test these kinds of paradigms, you also want to make sure that it is not reducing general reward. And so one of the ways to do that is to use sucrose as another rewarding solution outside of alcohol. So while it reduced alcohol consumption, it had no effect on sucrose. When we did the same thing in the central amygdala where there were no changes in IL-1 beta, we did not see any changes in ethanol consumption or the blood ethanol concentrations. Likewise, when we use another anti-inflammatory, interleukin-10, we saw that IL-10 reduces ethanol consumption in the BLA. That reduction in ethanol consumption also resulted in a decrease in BECs. Again, there was no change when we gave sucrose and no differences when we looked at giving it in the central amygdala. And so far, what we've been able to manipulate is really in the amygdala. But recently, we had a paper come out that shows that this dysregulation of the neuroimmune response is not isolated to the amygdala in terms of non-dependent binge alcohol consumption. And so we've gone back and we've looked at the hippocampus, and we see a similar story there. So we see that binge alcohol exposure actually increases IL-1 beta in the hippocampus and decreases IL-10. And then we also looked at specifically some glial changes. So looking at astrocytes, which has actually already been published. So we see an increase in astrocyte activation following multiple binge exposures in the hippocampus, as well as we've recently done some work looking at microglia, but that's not published yet. And so overall, our take-home message, or the take-home message for this presentation, is that excessive alcohol consumption induces neuroimmune responses. Those neuroimmune responses feed back and affect both biology and behavior. So specifically today, I only talked about behavioral paradigms, but we've also done things where we've manipulated the neuroimmune response to see whether it corrects for or decreases neurodegeneration. And so our hope is that therapies targeting the neuroimmune response may reduce drinking and therein reduce the damage associated with excessive alcohol consumption. With that being said, I'd like to thank my mentors, my previous mentors from my postdoctoral experience at UNC, as well as my graduate mentor at UK. The majority of the work that I showed today was actually done by some undergraduates at High Point University, by Drew Evans, Eva Greengrove, and Bella Grafassi. And I'm still building the lab. Actually just started at Central about 6 weeks ago now. And so I currently have a few undergrads who are signed on to work in the lab. None of this would be possible without support, financial support. And so some of this was supported by NIAAA through a UF54. And we recently got an award from the Alzheimer's Association that has also helped to further these projects.